Good morning and good afternoon to all. So I am Robin Joshi, working as a senior research investigator at Fitzgerald Lab in University of Pennsylvania. So I would like to thanks for giving me an opportunity to deliver a talk at this very prestigious occasion of India Metabolomics Symposium 2024 on shaping the future of precision medicine. So the title of my talk is Spatial Lipidomics and Proteomics of Atherosclerotic plaques in male and female hyperlipidemic mice. So my work is on mostly on spatial imaging mass spectrometry, that as how the disease progress. So what are the different lipids and what are the different proteins that we are able to identify, and so that on the spatial location of the tissue section. So as we know, the atherosclerosis is a risk factor of stroke, which is the one of main cause of mortality and disability worldwide. If you can see the artery, just can I move the pointer? Yeah. So it's like this is a normal artery, and when we as the disease progress, so there is a formation of the atherosclerotic plaque, and there is a when there is progress of the disease, it form around in the within the artery as it growth increases as progress of the disease, and it causes the blood clotting and mainly heart attack and some other disease, uh, um, uh, coronary artery disease formed. So so our main aim is to determine how different types of lipid present in these atherosclerotic plaques and as compared to the normal artery. So as we know, the men are more likely to present with sudden death and acute myocardial infarction, whereas women display a apical symptom of angina pectoris. Overall, plaque burden and the marker of inflammation appear to be greater in men as compared to women, which are predictive of cardiovascular event. So understanding of these feature of plaque stability, which have provided a framework for much of the work regarding for complexity mechanism that eventually lead to uh, atherosclerotic cardiovascular disease related event. So in the plaque formation, lipid play a very important role as the development and the progression of atherosclerosis, but still the involvement or nature of the uh, their how the lipids are involved, which is not fully understood. So it is very important to understand the sex specific uh, risk, stroke risk, which how the atherosclerosis is formed in the male and the female. So on the basis of this, there are some unanswered questions or aims in our present study. Uh, there are some of the unanswered questions, like to determine the hyperlipidemic influence of the spatial lipidomics and proteomic profile of atherosclerotic plaque in the hyperlipidemic mice. So we consider both the sexes, the male and female, on the Western high fat diet, and to analyze the plaque features, like what is the size of the plaque, what is their composition, and what is the morphology of the plaque, and what are the common or the different plaque features between men and women. And also we also investigate the it is very important to investigate the characteristic feature of the atherosclerosis as as the disease progress in the men and the women aorta. So and also to determine the unreveal the synthetic biosynthetic pathway of lipid as the disease progress. And the final question is how these lipids are synthesized. Are they are coming from what type of cells? Like, are they coming from T cells or macrophages and in the aortic plaques? So these are some of the unanswered questions. So to answer these questions, we use the spatial imaging mass spectrometry technique. So I use the DESI-MS, desorption electron spray ionization, or metric assisted laser desorption ionization, MALDI MSI. So in this, I'm just giving a small brief background of these spatial imaging techniques, like mass spectrometric imaging. Here we prepare the sample tissue by cutting tissue on the 10 micron section, and on the daisy or Mali, it goes to the, in case of Mali, it gives a laser and collect the uh, um, M by Z at pixel by pixel at particular time, and we generated a image M by Z spectra pixel by pixel, and with using different software analysis, we mapped all these pixels into form of images. So it gives the exact location of the M by Z, where these M by Z or where the lipid metabolite or protein present in tissue of the in the tissue. So this is the whole workflow that I have used in our study. So we use the mice model having athero and non-athero. We keep the take the male and female mice, 18, 16, and 32-week mice of high Western high fat diet, and take their extract their heart and cut their aorta using cryosectioning. And we made three different slides, one for DESI, one for MALDI, and one for HNE. So we use the HNE to map our atherosclerotic plaques with our imaging data. And with the unique feature, we are able to identify with the LCMS-MS for identifying and confirmation of the lipids using MSMS. And after that, we determine the spatial features of the lipids and proteins in our sample. And we use the Lipia and Bioman software for analysis. So Soon is one of our main project partner. He's working with the section cutting. Dan and Zineao, they are working with the data analysis of DESI, and Alim is supporting with the our DESI-MS analysis. So this is a small background about lipid classification. These are different types of lipid, like fatty acyl, glycerophospholipid, in which PC, PE, PI, and PS. These are main classes, and others are lyso, LPA, LPC, and LPG and LP. These are all monoacyl glycerophospholipids, and these are glycerophospholipid in which monoacyl glycerol, diacyl glycerol, and triacyl glycerol. And in the sphingolipid, the major classes are ceramides and sphingomyelin and cerebrosides, and sterol, in which cholesterol ester, stearyl glucosides. And in case of prenols, which are contain like glycan, phosphoglycan, and pyrophosphoglycans. So in our male and female heart section and the aorta, so we observe the different class of lipid in the positive mode and in the negative mode. In the positive mode, we observed lesser classes, in which we observed only 9 different classes, but in the negative mode, we observed more classes and more number of lipids. And when we compare with our in our samples, like 8-week to 16-week, and we found the 12 lipids which are showing upregulation, and 4 are downregulation with the significant cutoff value, and in which sphingomyelin LPC 18.1, sphingomyelin 26.5, and two triglyceride observed, which are showing high value uh, at the 16 week as compared to 8 week. And when we compared 32 weeks with the 8 weeks, and we found that this LPC 19, some diglyceride, sphingomyelins, triglyceride, LPC, these are showing up in the 32 weeks as compared to 18 weeks. The 32 weeks, we observed 37 lipids which are upregulated and 9 lipids which are showing the downregulation. So in the one of the previous study, they used the multi-imaging mass spectrometry, which revealed the special correlation in the mouse heart with the acute myocardial infarction, and they showed that uh, uh, they showed the some of the lipids in the positive mode, LPC showing the one of the uh, reason for the accumulation in the aorta for the myocardial infection. And in our data, we found that uh, all three groups we observed in 8, 16, and 32 weeks, all groups are different because of uh, dif- distinguished type of lipids as well as in their differential expression values. And with the VIP score, we found that if you can see critically in the 32 weeks, we observed high intensity of the lipids at 32 weeks, which is showing red color more in the 32 weeks as compared to 16 and 8 weeks. So it means like when we, as the disease progression or as the mice on the high fat diet, we increase the time. So there are accumulation of the more lipids in the 32 weeks. So in another study, they one of the authors they showed the accumulation of the sphingomyelins and ceramide phosphatase and sphingomyelins. They observed in the infract region of the aorta. So they showed that some of the sphingomyelin and ceramides, they are showing high intensity in the infract region. And they further show that the accumulation of sphingomyelin, which is also present on the periphery of the infract region, that can be correlated with the inflammatory cytokinin, and indeed that can induce the inflammatory response. So our imaging data showed that as we progress from 8 week to 16 week, this is the MALDI imaging data, and showing as we disease progress from 8 to 16 weeks. So these are different biological replicates we have used in the positive mode and in the negative mode. And the green color with the segmentation analysis, green color shows the athero around the lumen part of the aorta. It clearly shows that as we increase from 8 to 16 to 32 weeks, there is increase in the green color showing the accumulation of the lesion formation in the 32 week is more, and it is showing clearly in the positive as well as in the negative mode. It is clearly show at here in the 32 week showing very high lesion formation here. And some of the receiver operating characteristics, it's like a we compare the our two section like 8 to 32 weeks. These are top 15 feature we have identified using imaging mass spectrometry, and these are the how ROC means like how univariate measurement, how two sections are different in their intensity as well as in the uh, uh, eh, uh, you can say morphology as well as in their how different in their differential expression of the different type of the feature observed in the two section. So in one of our MALDI data like LPC 18 and LPC 18.0, these showed the when we go from 8 week to 16 to 32 weeks. So it is showing clearly the yellow region is showing higher intensity around the lumen part of the aorta. And you can see clearly from the intensity, it is clearly increasing trend in the LPC 18.1. And in case of LPC 18, it is also showing the high intensity at the 32 weeks as compared to 8 weeks. So this is also confirmed from H&E image of male mice. If you can see the H&E image overlapped with our imaging mass spectrometry data. So it is the 8 week of the aortic core of the aorta. So it is the 16 week. You can see the lesion formation is increasing, and at the 32 weeks, the this code is become more broader and lesion formation is high and lesion burden is high. And this is also confirmed from H&E as well as from the imaging analysis of mass spectrometry. So recently a paper comes that LPC and LPA 18.1, these are involved in the cardiovascular risk estimation. These are the two main important lipid that we that identified for the in the atrial architecture. So after that, we did the pathway enrichment analysis, and we took all the feature that we have identified in the lipids. So we found that the glycosphosphorlipid metabolism is the top pathway in which it is mapped with the KEG, and here red dot showing the how these pathway, how what are the different genes involved, and with the biopan software, we are able to identify what are the different type of gene involved in synthesis of these lipids. So with this biopan software, we found these some of the active classes of the lipid, how these I'm showing only active status of the lipid, how these lipids are converting, or how you can see the how these lipids are synthesizing from one class to another class. So and these are the genes involved, most active genes which are present in the data, uh, uh, which are present in the synthesis of one class of lipid into another. So these are the based on all predictive genes, based on all lipid that we identified in our spatial imaging data. So after that, till date, it was male story. Now, how the lesion burden increasing in the female mice. In the female mice, uh, uh, as we increase in the female mice, the lesion burden is less as compared to male. As we move from 8 to 16 to 32 weeks, and if we observe, if you can see the some of the important lipids. So in case of LPC 16.1, it is not showing any trend in the female mice. It is almost the intensity is same at the center of the lumen part of the aorta. But in case of LPC 16, which is clearly uh, um, show the increase in the intensity in the 32 weeks as compared to uh, 16 as to 8 weeks. And one thing I forget to tell you that data normalization is done with total line count. Secondly, we selected the specific area so that we can uh, take the intensity of only aortic region of the heart section, not the whole tissue. So these are the two main things that I forget to in the earlier slides. So after that, all these features are confirmed using untargeted lipidomic analysis. So we optimize our method from we extract the plaque and non-plaque area of the tissue, and with the help of LCMS-MS analysis, DD and DIM mode, we are able to identify the different types of lipid and able to confirm these lipid with the help of UJIL. Here we confirm the different type of lipid in imaging data. Uh, uh, After that lipidomic analysis, we optimize the workflow for proteomic analysis of the same slide that we optimize this method with Professor James in his lab in chemistry department. So we optimized some slide washing protocols for proteomic analysis and spray the tryptic digestion of the slide on the of the tissue in the on the sprayer. And after tryptic digestion, we spray the matrix and did the imaging, MALDI imaging analysis, and using skill lab software, we are able to identify the unique feature, and with the MSMS analysis, we are able to identify the different types of proteins in our uh, samples. So in case of these three sections, we observed that in the proteomic analysis, the lesion burden is also confirmed with the unique peptides which are observed in the atherosclerotic plaque in the aorta. So it is showing that those peptides are increasing in the uh, at the 32 weeks as compared to 16 and 8 weeks. And after that, in the proteomic.
Sorry, may I request you please conclude as we are running behind
Yes,
time?
yes, yes, yes. I'm concluding in 1 minute. Yeah. So in the proteomics workflow, the as we increase from 8 to 32 weeks, it's this cardiac limidin protein, which is uh, accumulated in the 8 weeks, and as we move from 8 to 32 week, it is decreasing. But here, this lamin B receptor protein, which is increasing in the 32 weeks as compared to 8 to 16 week, and this protein is mainly essential for the cholesterol biosynthesis. So this is the summary and major hypothesis that we drove from this data. Like male and female exhibit differences in the plaque composition as revealed by the MSI, and specifically we hypothesize that there is a high prevalence of the spatial lipid-rich plaques compared to females, and the distribution of the specific lipid or atherosclerotic plaque vary between male and female, and which is associated with the inflammation and plaque vulnerability, and the coronary artery of the female mice, which show the different spatial pattern compared to coronary artery in the males, and the progression is different over time. And the glycerophospholipid metabolism is strongly associated with the plaque formation in the male as compared to female. And PC, LPC, diglyceride, triglyceride, PE, sphingomyelin, these are the major classes noticed in the male mice. And these are some of the important genes which are involved in the lipid synthesis and their interconversion. And this is our team, and he's my main PI feature. And these are the people involved from collaborator from Alim's Wilji lab and the EJP James lab. And these are our funding sources from NIH. And I would like to thank you very much, Bijon and Pishu and Manoj and Monica, for this giving me an opportunity to uh, present my work and all by critic team. And I would like to thank University of Pennsylvania to allow me to deliver a talk on this. So thank you very much. If you have any question, please.